Fimbriae. We talk about Flagellae, Pili and Fimbriae. What do you mean by a flagellae? Flagellae is this makes bacteria motile layer, and the next form is the capsule. The slime layer form and the capsule form. Coming to the slime layer, it is very loose, thin, and helps in protection and attachment. Whereas coming to the capsule, it is very thick and tough, and also this capsule helps in the protection from the immune system. Like as they cause diseases in the other organisms, they help them in protecting from their immune system, and they prevent drying of the cell. So capsule is something that makes bacteria more pathogenic. What do you mean by pathogenic? It is more disease causing. So glycocalyx has majorly two forms. The outer membrane may be a slime layer or a capsule. Slime layer would be very thin, loose, and like this, a thin loose covering, which is majorly contributes in protection. But whereas coming to the capsule is very thick, tough outer covering, which majorly helps it from immune system of other organisms, and this capsule makes the bacteria more pathogenic and contributes to its disease causing capability. So after the glycocalyx, the next layer is our cell wall. So in the cell wall, what do this cell wall do? Cell wall majorly gives the shape to the bacterium. And it provides mechanical strength. Cell wall provides the mechanical strength and gives shape to the bacterium, and also it helps and prevent bacteria from bursting. It prevents the cell from bursting and collapsing due to osmotic pressure differences. So what do majorly cell wall do? Cell wall gives the shape and provides the mechanical strength to the bacteria, and helps in the prevents in the collapsing and bursting of the cell wall. Next we have the plasma membrane. Plasma membrane is the innermost layer of the cell envelope. This plasma membrane is selectively permeable. What do you mean by selectively permeable? That it does not allow all the components to enter inside the cell. It selects what to enter and what not to enter inside the cell. So what it does, it controls entry and exit. It controls entry and exit of the substances. This plasma membrane is structurally similar to the eukaryotic plasma membrane. Plasma membrane is structurally similar to the eukaryotic plasma membrane. Okay. So we have seen about the cell envelope and the 3 layers of cell envelope. Now let us see the modifications of the cell membrane. After this we will see the modifications. Coming to the modifications, the major modification of cell membrane that we have seen is the mesosome. Mesosome is the special membranous structure present in prokaryotes. It is the special membranous structure seen in prokaryotes. It is formed by the infolding of the plasma membrane. From where it is formed? It is formed from the infoldings of plasma membrane. Okay, so this infolding, it might be present as a vesicle, like a tubule, or like the lamella also. So this might be present as a vesicle, tubule, or a lamella. What are the functions of mesosome? Mesosome help in the cell wall formation. Mesosome helps in cell wall formation. It helps in DNA replication, and also the distribution of DNA to the daughter cells, in the respiration, secretion, and also in performing the enzymatic activities. So all these are the functions of the mesosome. What are the functions of mesosome? The cell wall formation, DNA replication, distribution of DNA to daughter cells, respiration, secretion, and enzymatic activities. Other thing to remember about mesosomes is that mesosomes are not seen in eukaryotic cells. Mesosomes are not seen in the eukaryotic cells. They are special membranous folding structures that are seen only in the prokaryotic cells. So after this, the other modification we talk about after mesosome is our chromatophore. We next talk about a chromatophore. What do you mean by a chromatophore? Chromatophore is present in cyanobacteria. Chromatophore is seen in cyanobacteria. It is a membranous extension. It is a membranous extension into the cytoplasm. This extends from the plasma membrane to cytoplasm. What do this contain? This contains pigments. This contains pigments and helps in the photosynthesis. Helps in photosynthesis. So the next modification of the chromatophore is we talk about the Flagella, Pili, and Fimbriae. We talk about Flagella, Pili, and Fimbriae. What do you mean by a Flagella? Flagella is this makes bacteria motile or non-motile. Motile bacteria possess flagella. When a bacteria is motile, it possesses a flagella. Okay. So structure of a bacterial flagellum, if you see the structure of this flagellum, it has a filament. What is the filament? Filament is the longest part outside the cell. That is the part that we are seeing outside. The long thread-like structure of flagellum is our filament. Coming to the hook. Hook is the part that connects the filament to the basal body. Hook is the part that connects the filament to the basal body. And then after the hook we have the basal body. Basal body is something that anchors the flagellum to the cell wall. This anchors flagellum to the cell wall. What is the major function of flagella? Coming to the function of flagella, its major function is to contribute in the movement. So if this is our bacterium, we have a basal body here, and then we have a filament. In between we have a structure called hook. This hook connects the basal body with the flagellum. So here are the three parts, that is the basal body and the hook. Hook connects the basal body with the filament. Filament is the longest part that is outside the cell. After the flagellae we have the pili and fimbriae. So what are pili and fimbriae? These are the surface structures. Both are the surface structures that is seen on the outer surface of the cell. They are not involved in motility. They are not involved in motility, unlike the flagellae. Flagellae helps in the motility of the bacteria. Coming to the pili, pili are long tubular structures. They are made of a special protein. And they help in conjugation and DNA transfer. We have seen what is conjugation. While the cells of bacteria are dividing, the DNA transfers from one bacteria to other bacteria. This conjugation is done by the separate tubular structures on the bacteria called pili. So pili are made of special proteins and they help in the conjugation and DNA transfer. After this we will see about Fimbriae. So what do these Fimbriae do? Fimbriae are short bristle-like fibers, like they are very short bristle-like fibers present on the surface of bacteria. They help bacteria to attach to the rocks. They help bacteria to attach to the rocks or to attach to the host tissues. So what do Fimbriae majorly do? Fimbriae majorly helps in the attachment of bacteria to other substances. So to other things, to attach the bacteria to other things, Fimbriae is majorly used. So what are the modifications that we have seen? We have seen the mesosome, that is the special modification of the plasma membrane. We have also seen the chromatophore. It's the modification present only in the cyanobacteria, and we have three other modifications called flagella, Pili and Fimbriae. So after this, we will now discuss about the ribosomes. Coming to the ribosomes. Ribosomes are the only organelles present in the prokaryotic cells as they are membrane bound. They are attached to the plasma membrane. They are majorly attached to the plasma membrane. Their size varies from 15 nanometers to 20 nanometers. They are made of, they have two subunits, that is they are made of two subunits, that is a 50S subunit and a 30S subunit. So both of these subunits together, they form a 70S ribosome. Okay, if a ribosome have two subunits named 50S subunit and 30S subunit, so these two subunits together they form a 70S ribosome. 70S ribosome. So what is the function of ribosome? As we already know that the ribosome helps in the protein synthesis. After ribosome, we also need to know about something called as polyribosome. Polyribosome is also called as polysome. What do you mean by a polysome or a polyribosome? That is, many ribosomes, what happens? Many ribosomes attached to a single mRNA. Many ribosomes attached to a single mRNA. These translate mRNA simultaneously and increase the efficiency of a protein synthesis. When you have a mRNA, many ribosomes are attached on the single mRNA. Now this is known as a polyribosome. So what do this polyribosome do? Polyribosome increase the efficiency of protein synthesis. Polyribosome increases the efficiency of protein synthesis. So now we have seen the ribosomes and polyribosomes. The next organelle we talk about is the inclusion bodies. So what do you mean by these inclusion bodies? These inclusion bodies, they are majorly the storage structures. They are the storage structures in cytoplasm. Okay, these inclusion bodies are not membrane bound. It is obvious as they are found in the prokaryotic cell. They are not membrane bound. So what is their major function? They are storage structures. They are seen in the cytoplasm. They store reserve food material. They store reserve.
Doubts stay uncleared. Hours of self-study. Yet biology still feels confusing. Because sometimes you don't need more lectures. You need personal guidance. At Crack Mentors, students get live personalized online one-on-one biology classes with dedicated faculty support, personal doubt solving, and focused improvement strategy.